In this video, I'm going to go through the electron transport chain. So this chain occurs through three levels of the mitochondria: the inner mitochondrial membrane space, the inner mitochondrial membrane, and the mitochondrial matrix. So let's start with the first stage. In the first stage, complex 1, which is the most left-hand green rectangle, pumps out 4 hydrogen ions into the inner mitochondrial membrane space, and in this process, an NADH becomes an NAD. In the second stage, coenzyme Q, coming from complex 2, transfers 2 electrons to complex 3. I would suggest that you don't learn the names of all the complexes. However, I would focus on this complex 2, which I've asterisked, because it's succinate dehydrogenase, which you would have seen in the TCA cycle if you had a look at it. So the connection between the electron transport chain and the TCA cycle is an important thing to know. So in complex 2, succinate dehydrogenase, in its process, an FADH becomes an FAD. Next up, we have complex 3. So cytosine C, or rather cytochrome C, that's the correct name. Cytochrome C, there's 2 of them in this case, and so complex 3 pumps out 4 hydrogen ions and also is aided by cytochrome C to transfer 2 electrons to complex 4. And there's 2 cytochrome Cs, which both transfer 2 electrons each. You don't have to know the full name of cytochrome C, you can just abbreviate it as cyt-C, but just something to note. Then we've got complex 4, which pumps out 2 hydrogen ions into the inner mitochondrial membrane space, and in this process, half an oxygen is converted to a water. So those are all the complexes of the electron transport chain. Let's move on to the enzymes. So we've got Atp synthase, which draws in 3 hydrogen ions from the inner mitochondrial membrane space, and in this process converts an ATP plus a pi, or a phosphate ion, to an ATP. So it's ATP synthase. It's making or synthesizing ATP. That's what this electron transport chain is all about. But we need to get this ATP out of the mitochondrial matrix. So to do this, we have ATP-ADP translocase, which brings in an ADP plus a hydrogen from the inner mitochondrial membrane space and pumps out a ATP into that space, which can then be used in other reactions. So we're just going to look at what the products are and summarize them. So the products of the electron transport chain are 1 water molecule and 10 protons in the inner mitochondrial membrane space. And approximately you need 3 hydrogen ions to make 1 ATP. Approximately, not exactly, it's actually a decimal point number, but if you would like to think of it that way, 3 hydrogen ions will make you 1 ATP, and that's why in the ATP synthase step we've got 3 hydrogens coming into the mitochondrial matrix space. So those are the products of the electron transport chain. Another really important thing to know is the regulation of the electron transport chain. So anything in green that I put down relieves the inhibition of the substance in red, which inhibits the complex of the electron transport chain. So let's get started. First of all, complex 1 is inhibited by rotenone, but this inhibition can be relieved by succinate. Next up we have complex 2, which doesn't have any significant regulation, but then we have complex 3, which can be inhibited by antimycin, and this inhibition can be relieved by ascorbate. Complex 4 is inhibited by cyanide and carbon monoxide. Then ATP synthase is inhibited by oligomycin. And finally, ATP-ADP translocase is inhibited by atractyl oside. So those are the key regulators of the electron transport chain, which you should remember. I'd also like to note that DNP can also inhibit the electron transport chain. DNP is dinitrophenol. That's a long name for it. So it inhibits this by bringing in many hydrogen ions and disrupting the ion gradient across the inner mitochondrial membrane. And formerly, DNP was used in a weight loss drug, but not anymore because it's incredibly dangerous. Disrupting the ion gradient across the electron transport chain was found to be quite dangerous, and it inhibits the production of ATP, which is giving energy to our body. So that's why it has now been removed as a weight loss drug. But just to note that DNP is an inhibitor of the electron transport chain. To help you out with remembering the regulation, my inhibition acronym is RACOA. So across from complex 1 to ATP-ADP translocase, excluding complex 2, I put the first letters in a little acronym to remember: RACOA, rotenone, antimycin, cyanide and carbon monoxide, oligomycin and atractyl oside. Those are the key things to know about the electron transport chain.